Dear students, after studying this module, you shall be able to know about the significance of radiochemical techniques, the basic principle and methodology of radiochemical techniques, and also you shall be able to know about the applications of these techniques, that is the radiochemical techniques. Isotopic and nuclear techniques play an important role in several aspects of everyday life and are a fundamental part of our socio-economic progress. Be it in medical diagnostics and treatment, in food security, in industrial process control, or in baggage screening at airports, these techniques serve basic human needs, fortify industrial affordability, or increase our understanding of nature and its processes. Radiochemistry is defined as the chemical study of radioactive elements, both naturally occurring and artificial, and their use in the study of chemical processes. The field of radiochemistry emphasizes on the analysis of sample for their radionuclide content. A number of methods are employed to find and detect the radio element of concern through chemical methods and sample measurement techniques. The field of radioanalytical chemistry was originally developed by Madame Marie Curie with contributions by Ernest Rutherford and Frederick Soddy. They established chemical separation and radiation measurement techniques on terrestrial radioactive substances. Activation methods were long held in remarkably extraordinary reverence in the field of trace evidence analysis. The first important breakthroughs in trace analysis of the elements, for example in semiconductor applications, accompanied the development of activation analysis, which was originally based on excitation with slow or fast neutrons, but later extended to comprehend the use of charged particles and photons as well. Methods based on nuclear reactions are restricted to laboratories specially equipped for handling radioactive substances, or radionuclides as we call them, under the management of trained personnel. Operationally, radiochemistry is defined by the activities of radiochemists, that is, nuclear analytical methods, the application of radionuclides in areas outside of chemistry such as medicine, the physics and chemistry of the radioelements, the physics as well as the chemistry of high activity level matter, and last, the radiotracer studies. Since the small amounts of material involved, the presence of radioactivity, which implies certain regulatory and safety concerns and the frequent need to deal with short-lived nuclei, these techniques are not the same as ordinary chemical techniques. Specialized techniques have evolved from the early part of the 20th century. These techniques are quite important, for they are often the key to a successful experiment, even though they may get scanty mention in descriptions of the experiment. Often, the successful application of these techniques, students, requires careful, painstaking attention to detail, frequent practice to develop the necessary manipulative skills, and a thorough knowledge of the underlying scientific principles of the subject. Radioanalytical chemistry spans many fields, including chemistry, physics, medicine, pharmacology, biology, ecology, hydrology, geology, forensics, atmospheric sciences, health protection, archaeology, and engineering as well. The applications include of radioanalytical techniques, forming and characterizing new elements, determining the age of materials, and creating radioactive reagents for specific tracer use in tissues and organs. The ongoing goal of radioanalytical researchers is to develop more radionuclides and lower their concentrations in people as well as the environment. The phenomenon of radioactivity presents to the analyst a number of specific properties that are characteristic of a particular radionuclide. These properties include the type and the energy of radiation involved, its half-life and the decay pattern of the radionuclide. An important point to note here is that the fact that a given nuclide may be radioactive does not in any way affect its chemical properties before radioactive emission takes place, and after detection its fate is of no consequence. In general, the chemical operations performed in radiochemistry are the standard techniques of analytical chemistry. Modern forensic science has to deal not only with homicides and other conventional type of crimes, but also with added comprehensive cautions such as trafficking of nuclear materials, surreptitious manufacture of weapons of mass destruction, amassing of prohibited drugs by state-controlled groups and warfare crimes. Forensic applications have always promoted the use of advanced analytical tools that can characterize materials found at the crime scenes. So, what are the general features of radiochemical analysis? Let us now discuss in detail this topic. Radiochemical analysis is based on two outstanding features of radioactivity. First, the high sensitivity and the ease of measurement of radioactive radiation. Second, the possibility of labeling compounds with radioactive tracer. Radionuclide techniques have often higher sensitivity than other analytical methods. The amounts of nuclides can be derived from the law of radioactive decay, which follows the first order kinetics. These amounts vary considerably, corresponding to the wide range of radioactive half-lives. Maximum of the commonly used nuclides half-lives range from several minutes to several years. So, the corresponding masses are extremely low. Radionuclides are often diluted with inactive isotopes, but specific activities, that is, activity per total mass of the element, are still very high. Since the background in nuclear spectroscopy is very low and sensitivity is quite high, activities as low as 0.2 becquerel can be readily detected. Analytical applications of radionuclide techniques rely on assumption that different isotopes of the same element exhibit the same properties in any macroscopic physical or chemical process, and that radioactive labeling does not influence the other properties of a chemical species. For analytical purposes, the radiotracer and the analyte must be present in the same chemical form. This is usually to achieve, but specialized preparative techniques may be necessary for radioactive labeling of more complex organic compounds. Radioanalytical chemistry offers a variety of accurate and highly sensitive methods. Even with the increased performance of the other trace analysis methods, including electrothermal atomic absorption spectroscopy or the spectrometry, atomic emission and fluorescence spectrometry, coupling methods, and high performance sample introduction in the organic and inorganic mass spectrometry, chromatographic preparations or separations, and neutron activation analysis, there are still a number of areas where radionuclide applications are unsurpassed. So let us now come to the importance of radiochemistry. Radionuclides are used in many subdivisions of analytical chemistry. The subdivision of major importance are radiotracers in methodological and pathway studies, isotope dilution analysis, which is referred to as IDA, radioimmunoassay, and neutron activation analysis, referred to as NAA. They are all especially suited to analyze the extremely small amounts of substances encountered in ultra-trace analysis or in trace analysis of microsamples. Over the past 2 decades, the emphasis has shifted from high detection power in routine analysis towards an independent approach, applying this high detection power to the development of analytical procedures and reference materials. Radiochemical methods for routine analysis have lost ground to other, primarily spectroscopic methods. Non-radiochemical methods often yield highly reproducible results, but may involve systematic errors. The need for increased reliability and analytical quality control has emphasized the usefulness of radiochemical methods for the certification of standard reference material, or the SRMs. Radioanalytical methods are often suitable for homogeneity testing and distribution analysis of traces in standard reference materials. Activation analysis, radio tracer techniques, and isotope dilution analysis are becoming increasingly important for the assessment of analytical quality. Now we shall study about the individuality of radiochemistry. Radiochemistry involves the application of basic ideas of inorganic, organic, physical and analytical chemistry to the manipulation of radioactive material. However, the need to manipulate radioactive materials imposes some special constraints and features upon these endeavors. There are several known radionuclides of this type, including 109 cadmium, 57 cobalt and 55 iron. These are the atomic masses, which have sufficiently long periods that they can be used as different energy sources. The small number of atoms involved in some radiochemical procedures can alter expected behavior. There are certain unique features to the chemical separations used in radiochemistry compared to those in ordinary analytical chemistry that are worth noting. First of all, high yields are not necessarily needed, provided the yields of the separation can be measured. Emphasis is placed on radioactive purity, expressed as decontamination factor, rather than chemical purity as in chemistry experiments. The success of a radiochemical experiment is especially dependent upon the source or target considerations. In many nuclear experiments, as much time has to be devoted to the target preparation as in comparison to the rest of the experiment. In order to produce applicable sources, different separation and source mounting techniques have to be applied. In many respects, the chemical separations of radioactive materials are very similar to the common analytical procedures. However, there are a few fundamental differences. One of these is the time factor, which is over and over again presented by the short half-lives of the species involved. In the case of very high activity levels, the chemical effects of the radiation, such as radiolysis of water and other solvents and heat effects, may affect the above procedures too. To attain satisfactory counting efficiency, it is obvious that the counting arrangement must be chosen very carefully. The radiations emitted by the substance and the available measuring equipment are among the determining factors. Special attention has to be paid to sources of low energy radiations if samples of very low activity need to be measured and when absolute disintegration rates have to be determined. Now let us study the advantages and disadvantages of radiochemical techniques, or sum it up in a way. Now, first of all, I'd like to take up the advantages of radiochemical techniques. It is a cost-effective technique It has element specificity and chemical site specificity. It displays high dynamic range in a single experimental run. Radionuclide blanks are often extraordinarily low, far below detectability. Next one is it can do multi-element analysis by simultaneous use of different tracer nuclides, often without separation. The quality and quantity of the signal producing decay are independent of the influences such as temperature, pressure, external fields, and the chemical environment of the nucleus. It locally resolves the identification. Prominent distribution detection, for example, from within an analytical decomposition apparatus, a growing plant, a thin layer chromatogram, or sections from the biological tissue or engineering materials. It has a lateral resolution as low as 10 picometer by autoradiography. It demonstrates predetermination of counting precision. This technique shows in vitro and in vivo with fast element detection, often from outside analytical system at comparable levels of sensitivity and accuracy as mentioned earlier too. And there are no interference with analysis by other methods, for example, non-destructive and conservative. Now we shall discuss the disadvantages of this method or these techniques. The synthesis of labeled compounds may be very difficult. Safety considerations are required to avoid incorporation of radiotoxic substances and radiation exposure. Specialized laboratory and equipment, qualified and skilled staff, disposal of radioactive waste have to be taken care of. Boron, aluminium, nitrogen and oxygen are of analytical interest, but no nuclides are available with half-lives longer than 10 minutes. Specific sources of errors are there due to radio tracers have to be controlled. There is no distinction between different chemical species without additional separation. In general also, sample material cannot be returned to normal use after analysis. So this probably are the biggest disadvantages of radiochemical techniques. In this module, we have taught you that radioanalytical chemistry covers the use of radioactive nuclides and nuclear radiation for analytical purposes, the development of radio reagent methods and further development of radioanalytical chemistry has established a range of analytical methods and techniques. Analytical applications of radionuclide techniques rely on assumption that different isotopes of the same element exhibit the same properties in any macroscopic physical or chemical processes. The success of a radiochemical experiment is especially dependent on the source or target considerations. In many nuclear experiments, as much time has to be devoted to the target preparation as to the rest of the experiment, as we told you. Radioactive measurements have associated problems such as the exposure of people working with them and of the treatment of waste materials, which means specific risk to the staff and the environment. Thus, we need very skilled personnel for carrying out these techniques. Radionuclide techniques often have higher sensitivity than other analytical methods. The amount of nuclides correlated to the activity of 1000 becquerel can be derived from the law of radioactive decay. The application of radioactive materials to the study of problems in biology, physics, chemistry and industry is growing rapidly. This is mainly because of the utility of radioisotopes as analytical tools. Lastly, radioactive methods of analysis are particularly useful for the detection and determination of trace quantities of materials and for the measurement of larger quantities in complicated systems or samples. Students, I hope with this all information on radiochemical techniques, you have found this module very interesting and informative.